So today we have the pleasure of having Dr. Hamilton O to present to us his recent work. And actually, he's a fellow currently in the Icahn School of Medicine at Mount Sinai. And he studies how the brain interacts with the rest of the body in a chronic psychological stress and depression. He also did his PhD from Stanford University. And I think this article came from that dissertation, which is looking at the organs age using large scale plasma proteomics, and identified through the CSF fluids and how to predict resilience and susceptibility to Alzheimer's disease. With that, I'll turn it out to Dr. O, and he can talk more about his study about omics and the brain aging. And thanks again for joining.
Yes, thank you, buddy. Hold on. Screen share. Okay. You guys see the full screen?
Yes.
Okay, great. All right. Yes. Yeah. Thank you, Fadi, for the invitation. It's a pleasure to share my work here. So yeah, I'll be talking about a couple of different studies that I did in my PhD, as well as some new unpublished work from my postdoc as well. But overall, the theme is really using the latest technologies in large-scale human proteomics. So proteomics in plasma and cerebrospinal fluid and accessible human fluids to get a window into the molecular changes with aging and cognitive decline in humans, and all in living people. And so the first part of the talk will be focused on understanding trying to get at aging. And really the motivation for this was that there's so many chronic diseases, and a lot of clinical medicine is focused on trying to treat diseases when they arise. But if we could understand what is actually happening, 50, 60 years before those diseases are developed. So what is that process of normal aging? What does that look like? Maybe we could get an understanding that will help us develop preventative medicines. And so really, we're interested in how we age, and are there ways to slow and prevent age-related diseases. This is obviously super interesting from a clinical perspective, but then also it's a really fundamental process of biology. And so there's many studies in animal models now showing in mice and worms and flies. Actually now in dogs, there's a company focused on longevity in dogs called Loyal. But basically there are these conserved mechanisms. And so there's lots of interesting work in this space, but we have this perspective. I mean, a lot of people know, but really the animal models are great, but they're not close, or many are, in many ways they're close, but in many ways they're not close to humans. And for example, just one of the major differences is if we think about how do humans die versus how do lab mice die. Humans die mostly from cardiovascular disease. Actually, lab mice are sort of resistant to cardiovascular disease, and they mostly die from cancer. They're bred that way. But humans, mostly cardiovascular, good amount of cancer, but also less respiratory diseases, digestive, neurological, and many others. And so we are interested in trying to understand this process specifically in humans. So how do humans age? And really we're excited to utilize the new latest technologies in blood proteomics, because the blood system is basically connected to every cell, tissue, organ in your body. And so then if you take the fluid fraction of the blood, you can measure all kinds of molecules, from proteins, which are the ones we were primarily interested in these studies, but also you can measure lipids and metabolites. And that can tell you, give you information about the physiological state of particular organs in the human body. So for example, when we go to the doctor and we get a blood test, one of the things we measure is cholesterol, or LDL and HDL and triglycerides and such. And these can give you a sense of how much your arteries may have been clogged over time, over your lifetime. In terms of proteins, there's a specific protein from the liver that is measured in the clinic called ALT and also AST. So these are liver enzymes that are indicative of acute liver damage. And so sort of just translating this and sort of going a step further, we thought, can we measure not just 10, 20 proteins that are measured in the doctor's office, but use the technologies to measure thousands of proteins from the blood to try and get an understanding of the physiological state and particularly the aging state of several different organs in the body. And so here we've used SomaLogic and Olink. So these are the two major human proteomics technologies that are probe-based. So they're basically high throughput, scaled up ELISA assays, similar in that sense. And so we measured roughly 5,000, 3,000 proteins across thousands of people. And now actually the technologies are even better. So you can measure around 5,000 to 11,000, depending on the platform. But when we started this study, so this was a major collaboration with co-first author Jared Rutledge, who was also a PhD student when we were both there. And so the first step was just so we had this data, all this plasma proteomics data. First thing we tried to do was map all of these thousands of proteins to specific organs of origin. So we tried to predict where they come from. For example, so the way we actually did this was, in brief, we looked at bulk RNA-seq atlases, like the GTEx atlas of different tissues, and we tried to identify organ-specific genes based on RNA gene expression. And then if we found an organ-specific gene, we then asked, is that protein encoded by that gene detected in the blood? And then we'd call that an organ-specific protein. And so we did this across all thousands of 5,000 proteins, found proteins that likely originate from the brain, heart, kidney, and so on. And this is the general picture of the number of organ-specific proteins that we could sort of confidently map to these different systems. And so brain surprisingly had the most number of unique organ-specific proteins. The liver was also really high. Liver makes sense because most proteins in the blood actually come from the liver if you're talking about the absolute concentration. But in terms of the unique number of protein names that you can identify, the brain has quite a lot. And so this was quite exciting because it suggested that maybe actually, for the first time, we could get a sense of not just, blood and liver aging, but also brain aging, which has been traditionally sort of an inaccessible tissue, mostly thought to be due to the blood-brain barrier. So then how do we actually go about quantifying the aging state based on these proteins? Well, we were inspired by some methods in the genetic space. So if you've made epigenetic blocks, those have more prominent ways to estimate biological age. And I'll go over them, explain them briefly. So this idea of quantifying biological age is, I think, really quite intuitive because we already do this all the time. And so I'll give you an example with the Karate Kid. Basically, if you know this movie, it's an old movie. This kid was mentored by his sensei. And actually, if you look at them now, or this was only a couple years ago, this is the Karate Kid now, and this is actually the sensei back in the movie, and so he's supposed to be old here, and he's supposed to be relatively young, but in terms of their actual age, the Karate Kid grown up is actually older than his sensei in the movie, but of course to us it seems like the grown-up Karate Kid still looks younger. I mean, to me, and maybe most people, maybe not everybody. And so, we intuitively say, oh, this person looks about this age based on the features on their skin, their eyes, and so on. And we can also just ask um, machine learning models to do this as well. So you could ask ChatGPT based on these photos, how old do you think they look based on these photos? And ChatGPT will say, the Karate Kid looks 50. He looks younger, even though he's actually older here. And so from here, then we can calculate something called an age gap, which is the deviation between their predicted age or the biological age and their actual age. So the Karate Kid has a negative age gap because he looks younger than he is, and Sensei looks older than he is, so he has a positive age gap. So we basically do this across not instead of using AI and ChatGPT to assign biological age based on an photo of a person. Actually more simple machine learning models to predict the age of people based on the levels of their proteins. And specifically, we're asking based on the levels of brain-specific proteins to estimate brain age, of kidney-specific proteins to estimate kidney age, and so on. So for each of these different 11 major organ systems that we looked at, we have an estimate of each person's, organ-specific biological age relative to their chronological age. And so, for example, this is the kind of plot that you would end up with, where you have their predicted ages versus chronological ages. Each dot is a unique individual. And so this person has a relatively high organ age gap, and this person has a negative organ age gap. And so this was the general idea of the study, and this is a major collaborative effort across many cohorts. So these big plasma proteomics data are really quite expensive, and so this definitely, huge consortiums working together to generate this kind of data. And so we worked with Carlos Cruchaga from Washington University, St. Louis, Neil Barsley from New York, Einstein School of Medicine, Sophia also from Einstein, and so our collaborators at Stanford, Anthony and Beth. And so we had a training cohort where we would train the models, the organ-specific aging models, and then we would test them across all of these independent cohorts. And so one of the first questions we asked was, how unique are the organ age gaps? Is it true that actually organs age at different rates? Sort of based on the epidemiological data that people have different incidences of various organ-specific diseases. And one of the first things we found is that organ age gaps are actually pretty distinct across, even within an individual. So here's just 3 examples. So each column here is a person. In the middle, you can see the gray circle, which is actually their conventional age gap. So conventional meaning this is an aging model that doesn't use any organ-specific proteins. It just looks unbiasedly at the whole plasma proteome to derive a biological age estimate. And so this is what's typically done in the field. And you can see that all 3 of these people have the same conventional age gap. So the same biological aging score, if you will, based on baseline models. But the organ ages are really quite distinct. So this first person has young organs across the board, and person 3 has old organs. And this is just showing an example of a couple individuals. But then if you look at pairwise correlations across all individuals for each pair of organ age gaps, you can see that there is somewhat of a correlation, but it's actually pretty mild. So the average correlation is really only, R equals 0.25 between any pair of organs. And so this suggested that organs really might be actually aging at different rates. Another question, so sort of following up on this, is that if people are aging differently, can we actually identify really unique types of agers that really are aging only in one organ compared to the rest? And we also found examples of this. And so here's again three different individuals. Now I'm plotting their z-scored age gap. So a z-score of 2 means like the top 96th percentile, I think. And so these are really quite rare individuals. But you can actually identify these people who are really old in just one organ, kidney or heart, versus old in almost all organs. And if you plot all of these different extreme ages, you can actually really pull them out. So you can identify individuals that are aging primarily in one organ versus multiple organs across the population. And this was roughly 25% of people could be classified into one of these groups. And so it suggests, yeah, it again suggests that people are aging differently. And not only that, you can actually identify people who are aging sort of in a more extreme way in a particular organ compared to the rest of the people. Um, and and so, this was in our first study, which was a sample size of roughly 5,000, I think. But so we weren't able to ask, does the extreme ager population, so there's like 2%, it's a really relatively small sample size, does this actually have implications for a person's future health outcomes? And so we were excited to analyze the UK Biobank data set, which is a relatively new publicly released data set of 50,000 individuals, 3,000 plasma proteins measured per individual. And the UK Biobank is a longitudinal population cohort. And so they have linked electronic medical records over about 15 to 17 years. And so here we could actually ask, do these people who are kidney agers versus heart agers, multi-organ agers, actually have differences in future health outcomes? And so one of the first things we looked at was mortality. Sort of the gold standard in the aging field is, how long are people living? And what we found is that compared to these, so this is looking at survival over around 15 years. And you can see in the gray here is the normal agers. And now we're looking at the extreme agers. So we're looking at extreme agers with 2 to 4 aged organs. That dramatically increases risk, roughly 2 times increased risk. And then if you have 5 to 7 or 8+ aged organs, that's again, 5 times to 9 times increased risk. So the extreme agers, it actually does seem to be a biologically meaningful classification, as in that it's not just sort of like outliers based on noise, but there's actually, biologically relevant, maybe clinically relevant information in these biological age estimates.
Which one, or who say you rather go to another JUCO college? Isn't that what you said? Which one? Yeah, so which one are you?
Okay, I'll just continue. Sorry, I didn't understand if that was a question.
No, it's okay. Sorry, that was Dr. Paula. She may be doing clinical service there. Sorry about that.
Okay. Yeah. So was I? So yeah, having multiple aged organs progressively increases risk, and actually what we found is that having a young brain and young immune system was particularly beneficial, and actually uniquely beneficial in being associated with longer lifespan compared to the normal aging population. Again, this was really unique. So we actually didn't find that if you had a young heart only, a young kidney only, that actually didn't associate with longer life. Really, it was the young brain and the young immune system. So potentially pointing to those systems as really key for a wide range of functions and maybe for longevity in humans. There are also many other associations between organ ages and organ-specific diseases. So heart failure associated with increased heart age, and many others. But I think the strongest one was actually, and maybe most relevant here, is that brain age was predictive of future Alzheimer's disease over 15, 17 years. And again, not only was having an old brain indicative of increased risk, but actually having a young brain was associated also with dramatically reduced risk, 75% reduced risk compared to sort of the average population. Suggesting that we really are And so actually, so this was a unique association with the brain. So if you looked at all the other organs versus future Alzheimer's disease, the effect was really not that strong. I think only a couple were even slightly significant. And so it suggests that, maybe for the first time we can actually begin to measure the normal aging process of the brain, with a blood test. Yeah, and then if also not only are we like getting an estimate of the brain age, we also want to understand what is actually the molecular processes that might be changing with brain aging. And so if you look at the proteins that are in this sort of brain aging model, what you pull out are proteins from various different cell types of the brain, including astrocytes, neurons, and actually quite a big enrichment in oligodendrocytes and oligodendrocyte precursor cells, or OPCs. Suggesting that we're getting hints at what processes might actually be changing. GFAP is a known a marker of astrocyte reactivity or inflammation. And NEFL, neurofilament, is a known marker of axon degeneration. But many others, sort of less understood proteins like MOG, which is myelin oligodendrocyte glycoprotein, might be relevant. And a marker of sort of myelin degeneration potentially, and BCAN or brevican is a extracellular matrix protein. And so potentially these proteins in the blood might be indicative of the structural support in the brain that's changing with age. And so to summarize, we can now estimate the biological age of individual organs by measuring organ-derived proteins in the blood. And so I think this is really quite a big step forward because, previously it's been possible, to study or at least get measures of the function of particular organs, like, if you want to measure blood pressure or respiratory VO2 max and volume, you can get a measure of the function at that moment in time. But then these proteins might actually be indicative of not just function now, but function in the future. And so we really a molecular measure, one of the first molecular measures of specific organs, and in particular the brain and the aging state. Two is that biological age is actually associated with future organ disease and mortality. So it's not just some number that's generated by a model and who knows if it actually has any relevant information. We actually thoroughly show that these age gaps have pretty strong associations with future mortality and disease, actually. So if you want to predict mortality, the organ ages are more predictive than what is currently measured in clinical biochemistry. And so there's definitely meaningful information here. And third, I think is a future direction is now it's going to be really exciting to ask which interventions can actually maybe slow or accelerate organ aging. And so this is, I think, a major goal in the longevity field is to identify new drugs, new compounds that can extend health span and lifespan. But to do these trials, it takes obviously a really long time if you want to see how long someone is going to live. But if you can show that a drug reduces the aging metrics across the board, maybe that could be an early indicator. Okay. And then, lastly, can the proteins we measure to monitor aging also be drug targets? As I mentioned before, we're also getting a molecular understanding of what the aging process might actually look like in humans. And so there'll be many more studies to actually figure out the mechanisms and to see if any of them are druggable. But I think it's a promising early look. Okay, so for the second part of the talk, I'm going to be focusing more on neurodegenerative diseases, especially the dementias, Alzheimer's dementia, as well as frontotemporal dementia. So a little bit of background, Alzheimer's disease is a really devastating age-related dementia. And I think these these self-portraits by William Otermel and really capture what it's like. It's not just loss in memory, but it's a loss in self-identity and just normal functioning. That the risk dramatically increases as you age. Over 100 years ago, Alois Alzheimer's identified what we now know of as amyloid plaques and tau tangles from the brains of patients with dementia. But really, these are now known as the key hallmark pathologies of Alzheimer's disease. And what's interesting is that they actually change, several decades. So amyloid is thought to accumulate around 2 decades before any signs of cognitive decline, and tau starts accumulating maybe a decade or less before cognitive decline and sort of drastically increases as dementia progresses. And so these are measurable in plasma. You can measure the pathogenic forms of amyloid and tau to pretty strong. Amyloid, at least, very well. Tau, it's getting, the biomarkers are getting there, but essentially you can measure and monitor these processes in living people in sort of a non-invasive or minimally invasive way. But what we found based on these data over the years is that actually Alzheimer's is really heterogeneous, and that these pathologies are not the only things that explain what's going on in Alzheimer's disease and the dementia. So for example, there's these are all the age of onset for Alzheimer's disease is super variable. It's mostly, late onset condition of after age 65, but there's also early onset cases that are sporadic, not due to familial mutations. Some people also live until 100 years old with actually no Alzheimer's disease. So there's a ton of heterogeneity, and the biomarkers don't explain it. And so here we're also looking at people who have mild cognitive impairment, so sort of early clinical decline, but not quite dementia. And the rates of cognitive decline are, again, really drastically variable, even if people have very similar levels of amyloid. So some people live to 100 years old and never develop dementia, even though they have Alzheimer's pathology. So even they might have amyloid and actually some tau as well, but they don't show any dementia. So it's clear that it's not just these two pathological protein aggregates that are responsible for dementia. And so we really sought to understand, are there other factors that can actually explain why some people get Alzheimer's disease dementia and others might have Alzheimer's disease pathology but no dementia? And really, we are interested in profiling proteins, again, with the sort of same technologies in large-scale proteomics to profile proteins in human cerebrospinal fluid to reveal what proteins might actually be most predictive of dementia, independent of those pathologies. So this was, again, a major collaboration, again, mostly with Carlos Cruchaga, who's really a pioneer of proteomics in the brain in the aging space. And then also, again, with my PhD advisor, Tony Weiss-Correa, who again, also leading the way in proteomics for neurological and aging conditions. And then also with Ville Leinonen in Finland, who's also studying a unique cases of Alzheimer's disease of IMPH, which is idiopathic. I forget what it stands for, but it's a condition where your ventricles there's too much pressure in your ventricles, and actually you have to that actually has increased risk of Alzheimer's. So we basically ran proteomics across all of these cohorts from sporadic Alzheimer's to genetic Alzheimer's as well, actually. So familial or autosomal dominant Alzheimer's disease in the Diane cohort. In these samples, we had not just the CSF proteomics, but we also had measures of amyloid and tau pathology from biomarkers and cognitive scoring. And in particular, we had longitudinal cognitive metrics across most individuals. So we could ask, what are the proteins that actually correlate with cognitive impairment independent of amyloid and tau Alzheimer's pathologies? And so what we saw from this kind of analysis was that there's actually tons of proteins that differentially are differentially abundant with cognitive impairment independent of amyloid and tau. Here we used the CSF p-tau 181 amyloid beta 42 ratio, which is a gold standard measure of amyloid and sort of early tau, but mostly amyloid pathology. And what we found is that if you look at the biological pathways of these differentially abundant proteins, it primarily changes in synaptic function, synapses. Again, potentially suggesting that actually we might be measuring synaptic dysfunction, synapse loss based on these protein levels. And synapse loss is actually a known, really strong histological correlate of dementia staging. Okay, so we had all of these differentially abundant proteins, particularly in the synaptic pathway, and so we wondered, could we make, a multivariate score based on all of these proteins? So in a way similar to the way we measure organ aging, where we say, let's combine the information from 200 brain-derived proteins to develop a brain age estimate. Here, we say, can we take the 700 or so differentially abundant synaptic proteins in cerebrospinal fluid and make sort of a synaptic score of cognitive impairment? And what we found was that the machine learning model basically only needs 2 proteins. This is based on recursive feature elimination, so it sort of almost plateaus at 2 proteins. And what it says is that it's essentially the ratio between YWJG, also known as 1433 gamma, and MPTX2, which is really strongly correlated with cognitive impairment across every cohort that we have. You can see the cognitive impairment scores. So from no cognitive impairment to slight, all the way to moderate and severe, increasingly associated with the synaptic ratio, also noted here as a synapse signature, across every cohort, including the DIAN, which is the familial Alzheimer's disease. Suggesting that here we have a really strong biomarker of cognitive impairment with only 2 proteins. What's key here is that this ratio is correlated with cognitive impairment independent of p-tau A beta, so independent of AD pathology. So this is if you look at the people on the sort of the right side, everyone here is amyloid positive, T1 positive, tau, early tau positive. So everyone here has enough Alzheimer's pathology to be diagnosed, at least based on their biomarkers, to have some sort of Alzheimer's disease, some level of Alzheimer's disease. And you can see among these people that actually it's the synaptic ratio that separates out who's cognitively normal versus who has dementia among the people with pathology. And so this was really quite exciting because we're going beyond the gold standard in the field and now have a really strong correlate of the actual cognitive function. We were curious how early in the disease process these synaptic proteins change. And so we looked at the normal aging process as well, because again, my background was studying aging. And so what was interesting is that the synaptic ratio also changes with age and normal aging really early on. So this is, the earliest we can measure, which is basically 18 years old when you're an adult. It actually steadily increases and sort of more rapidly increases in late life, compared to the Alzheimer's biomarkers, where it's almost pretty much flat until at some point, your 50 years of aging creates a state that allows these disease pathology to start accumulating. Whereas actually the synaptic ratio is potentially something that is sort of changing throughout our entire lives. This is reminded me of a study of non-human primates where they they characterized, what are the changes in neurons with just normal aging in non-human primates. And what they found was that actually it's the synapses that are changing the most. And so there's not raw sort of, or not overt loss of neurons or neurodegeneration, so to speak, but it's actually sort of more nuanced changes in the synaptic structure and synapse number that is majorly changing with the normal aging process, and also highly correlated with cognitive function in these non-human primates. And so again, I thought, maybe this ratio is not just capturing the dementia progression, but also really early. And to study this, sort of the links between the aging process and dementia, we looked at this, the autosomal dominant AD cohort, so the familial AD cohort. And what you can actually see is that the synaptic ratio, the change with age is faster in people who have mutations, for early onset mutations for Alzheimer's disease, but actually don't have any symptoms yet. So everyone here is cognitively normal, but in the red, you can see these are people who have the mutations compared to the gray is no mutation. So they seem to be aging faster, or the synapses are aging faster. And even in familial AD, you can actually separate, depending on the mutation you have, you can estimate when this person's going to get Alzheimer's disease. And so basically, if you have an estimated age of onset of around 30 years old, the synaptic aging rate, or so the slope with age is really high compared to if you have a mutation that's not as severe, where your estimated age of onset is around 60. And sort of scales. So the more severe your mutation, the faster this synaptic ratio changes with age. And then lastly, 8.4 also, the strongest genetic risk factor for sporadic Alzheimer's disease, also shows a slightly increased slope in the change with age, suggesting that these Alzheimer's might be aggravating a normal synaptic aging process. Okay, what was key is not just measuring changes with age or changes with cognition cross-sectionally, but being able to actually predict future changes in cognitive function over time. And so luckily we had access to these cohorts, amazing longitudinal cohorts where we had the proteomics and biomarkers, but also 15 years of annual cognitive testing. And so we can ask, does a baseline synaptic ratio measurement predict who's going to actually end up with dementia in the future? And so that is indeed what we found. And so basically, if we look at the A plus T plus, so again, people with can be diagnosed with Alzheimer's disease based on their pathology levels. And they have MCI, so mild cognitive impairment, so sort of right before dementia. It's the ratio groups that really separate out who is actually going to be relatively stable versus who's going to get dementia in the near future. And then looking at even earlier in the process, so looking at people who have Alzheimer's pathology, but no cognitive impairment at all. So these are, clinically indistinguishable from a normal age person. Again, we can separate out who's going to be cognitively normal over time versus who develops mild cognitive impairment or dementia within 5 years. So this is really quite promising, and it suggests that we have new measurements that can actually predict future outcomes beyond and independent of clinical gold standards. So we're also, of course, interested in trying to make a plasma proteomic surrogate of this. If we could actually measure these two proteins in the blood, that would be incredibly game-changing, because blood is so much more accessible than cerebrospinal fluid. Unfortunately, the two proteins, so cerebrospinal fluid levels of these two proteins are not at all correlated with plasma levels of these two proteins. And it's probably because these two proteins are expressed across many cells and tissues in the body, so that plasma levels are diluted out when you measure the plasma. But we could actually, using other proteins, at least partially recapitulate this CSF ratio. So we actually could make at least a signature that was really strongly correlated with cross-sectional cognitive impairment and dementia severity across, several cohorts. Actually, this is a pretty big sample of, I think, 7,000 or so, maybe 11,000 in total. And it could also predict future changes in cognitive outcomes, though not as strongly as the synaptic ratio. So I think as the proteomics technologies expand, so the more proteins we measure, and maybe if we can detect particular proteoforms that are linked to the CSF ratio, we might be able to translate this into a plasma biomarker as well. And now, just really quickly, I'm showing you some unpublished data on now frontotemporal dementia. And so actually it was shown that these two proteins from the synaptic ratio, the YWHAG and MPTX2, change with symptomatic frontotemporal dementia, or FTD. And so we were interested in investigating the ratio of these two proteins across FTD in symptomatic stages, but also pre-symptomatic stages and in the earliest stages, to see if how early do these proteins change in FTD, and can they also be good biomarkers of future changes in cognitive function. So this is a collaboration with Rowan Saloner and Adam Boxer from UCSF, who lead many of the sort of the most deeply phenotyped FTD cohorts, and as well as Henrik Zetterberg. So actually, first here is data on Alzheimer's disease. So actually switching back to Alzheimer's disease for a second, what's quite interesting is that the CSF synaptic ratio is really strongly correlated with, so CDR sum is a clinical dementia score, it's a dementia score, and it's more strongly, so the correlation between those two is stronger than tau PET versus the dementia score. So tau PET is really the gold standard for tau tangle pathology in Alzheimer's disease, and actually we're showing that our synaptic ratio is actually even more strongly associated with cognitive impairment than tau PET, which is quite remarkable. It actually fully mediates the association between tau PET and dementia severity, suggesting that tau PET or tau tangles might actually, you basically need the synaptic dysfunction downstream of tau tangles to actually have that cognitive decline. And then as well as it's also possible that even if you don't have lots of tau tangles, you might have lots of synaptic dysfunction driven maybe by amyloid or soluble tau that's more correlated with the clinical decline. Okay, and then back to FTD, we saw that this synaptic ratio is elevated and sporadic, as well as familial FTD to a similar degree as Alzheimer's disease. So here's one cohort at UCSF, the Memory and Aging Center, cognitively unimpaired individuals, FTD, symptomatic Alzheimer's disease. This is actually motor neuron disease. So people with a neurodegenerative disease but no dementia. And so the ratio is actually quite low here. So probably linked to dementia and cognitive function. And it's also increased in familial FTD across various different mutation groups. So familial FTD, there's mutations in either C9orf72, GRN, or MAPT. So mutations in those three genes can cause familial frontotemporal dementia. And so even across all of these cases, you see that this synaptic ratio is increasing with especially strongly at the symptomatic stage, but also slightly changing in the pre-symptomatic stage. And I think this is probably the most interesting, is that if you look at people in the pre-symptomatic stage in familial FTD, we asked, could this ratio actually predict who goes on to develop the synaptic ratio versus plasma neurofilament? And you can see the red are the people that end up developing symptoms within 8 years. So you can see basically everyone who develops symptoms is double positive for both biomarkers. And I think this shows it pretty nicely, where it really, having high levels of not just neurofilament, but also the synaptic ratio dramatically enriches for the people that are going to convert into symptomatic FTD among pre-symptomatic carriers. So again, I think a really promising biomarker for across dementias, especially early stage. So in summary, the synaptic ratio, to our knowledge, is the strongest molecular biomarker correlate of dementia severity, and not just in AD, but also FTD. It predicts future cognitive outcomes among people with pathology, Alzheimer's pathology, and independent of and beyond gold standard biomarkers. And combined with plasma neurofilament, it really strongly predicts who's going to convert from pre-symptomatic to symptomatic familial FTD. Some future directions and implications. It suggests that the synaptic dysfunction is likely really an early, not late stage pathological event in AD. There is some recognition of this, but I think basically the biomarkers haven't been, no good synaptic biomarkers have been found, I think, other than maybe these two proteins as a ratio that really show this to be true in humans. So we have some pretty strong evidence of this. MPTX2 may be a promising drug target for cognitive aging and dementia broadly. There's many studies actually in animal models showing that the function of this protein, it prevents sort of excitotoxicity and actually has benefits if you overexpress it in the Alzheimer's mouse models or actually even aging rats. And lastly, the synaptic ratio, I think, has really strong potential utility in Alzheimer's and FTD clinical trials, and potentially other neurodegenerative dementias as well. Okay, that's everything. That's a lot, but yeah, this was a huge effort. Many institutions. I'd like to thank my PhD advisor Tony Weiss-Correa, co-authors, as well as some major collaborators, including Carlos Cruchaga, Rowan Saloner, and Adam Boxer, and many others. And then, of course, my postdoc advisors for hosting me now. Thank you. Happy to take any questions.
Maybe I'll go first, since I don't see anybody jumping in ahead of me. I just thank you so much. I think this is so fascinating, and I really thank you for your time presenting here. You know, and there's so many questions, but I think the thing that I'm wondered about is the proteins in the blood that are showing you the aging brain. You know, I didn't see, is there a panel of them that you showed, like, for instance, NFL and MOG, you know, and markers overall for, you know, for different cell types in the brain. And I was just wondering, is there a full list, or are these the top ones that you're showing that somehow are highly associated with the aging brain?
Yeah, it's a good point. So these are essentially the top proteins, but the model is made up of around 50 is my guess. But I would say probably so the model actually gives the ranked list of proteins, but also their weights. And the most highly weighted proteins, you probably only need around 10 or 15 or so to sort of capture most of the signal. And it's these proteins, but also a couple other ones. They're in the paper and in the tables as well, if you're interested.
Got it. Because I was just wondering, because I mean, it's in following the stroke literature, you know, we haven't been very successful in trying to find proteins released from the brain. Let's say in the early stages of an acute stroke. We've all been really interested in that topic now for 25, 30 years, and it's GFAP always comes out, and
right.
You know, but we haven't seen a lot of other proteins that have been found to be specific to the brain, you know, and really not being released by other organs, which is obviously one of the issues you have here, I guess, you know, when you're trying to convert your CSF markers over to blood for the synaptic protein dysfunction.
Yeah.
So I was just curious what other proteins, but what you're saying is the highest ranked ones are the ones that you have listed here on the slide.
Yeah. I think also maybe it's like the technologies to measure brain proteins have gotten so much better, even just in the last like 5 years, that it might also like definitely not, most of these aren't detectable with mass spec, for example. And so it
How did you
might,
measure them? How did you measure them? Is it the Alamar panel?
yeah, so Alamar measures, I think, a good amount of these. This
Mhm.
was from Olink data.
Okay.
But actually SomaLogic doesn't measure a lot of these, or not that it doesn't measure, but SomaLogic and Olink, they're all probe based, so they're likely measuring different regions or epitopes of the protein. And so actually, if you look at the correlation between SomaLogic and Olink, it's like bimodal, where some proteins have really strong correlations, others have almost no correlation. And it might just be that they're measuring different proteoforms.
Yep.
But for these proteins specifically, I think these are the more uncorrelated proteins between SomaLogic and Olink. So the signal is much stronger in Olink for these.
Okay.
Yeah. And Alamar and Olink should be similar, I think. So they're both dual antibody based. Might even be using the same ones.
Okay, so I think I have like 1, 2 questions. So first of all, a great presentation, really, as we really enjoyed this presentation. So one question, so you mentioned synaptic, neurons, like the marker that comes from CSF, but can you differentiate which synapse is coming from? Like, is it axonal or dendritic?
Yeah. No,
Yeah.
so I mean they're expressed in both excitatory and inhibitory neurons. And then axonal versus dendritic is another. Yeah, I don't know how we could do that. But yeah, we haven't been able to get that level of resolution for sure. But I mean it's hard to say, because actually MPTX2 in animal models has been almost exclusively studied in neurons. And in those settings, it's been shown that it's expressed mostly by excitatory neurons. Or actually, I think it's also expressed in inhibitory, but in terms of the function, it's secreted by excitatory neurons and binds to receptors on PV interneurons. So the PV inhibitory neurons, and there's a very specific like relationship there. But actually in humans, the MPTX2 is also expressed by the oligo lineage, I think. And so, yeah, no, I think it's super unclear still, like what is actually going on in the human brain with these proteins. I mean,
Yeah.
with most proteins. Yeah.
Yeah. So the second question actually, so you measured all these proteins at the one time point, correct? And then you correlate with the age of the participant.
Yeah.
There's no 2-point measurement of the actual proteomics, correct?
Right. Most of it is one time point. There's actually a couple cohorts with longitudinal CSF data, and there's so the GNPC, if you've heard of that, it's the Global Neurodegeneration Proteomics Consortium. They just had like a release of like 4 papers in Nature Medicine all together, because it's basically a collection of like 30 different cohorts, like all putting their proteomics data together for neurodegenerative diseases. And in that cohort, there's an anonymous cohort that has longitudinal CSF over 5 years. And so the ratio also changes like longitudinally with aging in that. We haven't published it, but I just looked.
Thanks. Thank you again. Great. Okay. Apparently, there's no other questions. Probably yes.
I saw a question from Seema. It looked like she was wondering how the profiles would change in these patients that are now being treated with anti-amyloid antibody infusions.
Yeah, yeah. I mean, we would love to know. You know, There were some indications that the people who benefited most from anti-amyloid had low levels of tau PET. So since tau PET is sort of like a more late stage marker, it could suggest that you need to intervene with anti-amyloid earlier. This was in some of the more early anti-amyloid trials in the mild dementia, mild cognitive impairment stage. But it's also possible that the synaptic ratio could explain like who also responds versus who doesn't. And maybe you need maybe if your synapses are really dysfunctional, it doesn't matter if you reduce amyloid. Maybe it's too late also by then. So, I mean, I'd be really curious to look at that. I think most, yeah, the companies probably have some access to CSF from those trials because I think they actually screened for presence of amyloid in CSF because the plasma biomarkers weren't there at the time. But I don't know if they probably won't. They probably won't look at it. But in terms of like the latest data, I think, yeah, most people aren't collecting CSF anymore. So that's, I'd say, the major challenge is that the plasma biomarkers for amyloid and actually even tau are getting so good that yeah, and people don't want to get a lumbar puncture in the United States. So not many companies are collecting CSF from their clinical trials. But
Yeah.
I'd love to know.
Which is precisely why when you started talking about the synaptic markers, I was getting excited until Fadi told me on text, no, this is CSF, not blood.
Yeah, the blood is much harder. So I think, yeah, if we can basically find the signal in blood, that would be way bigger and more impactful. But I think the CSF is maybe a good first step in that we now have two proteins at least that we know are changing in the brain and CSF. And now maybe we can actually start to try and find particular proteoforms that are changing in the blood. So maybe the kind of like the development of the tau antibodies or like the tau tangle biomarkers in blood, we could potentially like pull down these proteins from blood and then do mass spec to see if there are particular proteoforms linked to the CSF and to the brain. Because maybe if you look at like the bulk signal, it's diluted from all the other proteins sources.
Yeah, and I wondered if that's what you were going to be doing. If you're in the lab and the PI, we're going to go in that direction.
Yeah, we're probably going to do it, but yeah, I mean, I would. It's really tricky. And I have a lot of projects, so I'm trying to balance everything. I would love to see this go forward in some way. So, if I don't know if you guys are interested or if you know anyone that's interested, I'd be happy to talk more. We're fingers crossed we're like likely going to get funding to do it. But it's more about execution, which is again pretty hard.
Yeah. And then I see an interesting question, from Deborah Clegg, who's VP of Research in Texas Tech, El Paso. And she was just wondering what other sources of tissue, she mentioned sputum, for example. It's a very interesting question.
Spit?
Well, it's more than that. I mean, you got to get down deeper to the respiratory tract to get actual sputum samples.
Oh. I don't know. I doubt it, but maybe the sputum samples would be good for. I just don't see how proteins from the brain would end up in the sputum. Maybe it's possible, but yeah, I don't know. Probably just have to see.
Alamar has been able to distinguish like the protein, some of the tau, that comes from the brain and that which is from all the other organs. Is that something that you could work with them to do and collaborate on, or is that you're going to be doing that in the lab yourself?
This is not for the sputum, right? This is for like
Oh
the
yeah, sorry, I was back to the blood.
back to biomarker. Yeah.
Yeah.
Yeah. So Alamar, I don't think Alamar develops their own antibodies.
Okay.
I
They just take them from someone who's
I think
developed
so.
them.
I
Okay.
think so. I think they take most of the established ones. So, I think Olink and SomaLogic are in a race to expand their menu, whereas Alamar is trying to deliver very sort of like tight-knit, verified menus.
Panels.
Yeah, panels. So, in terms of like, yeah, more risky new biomarker discovery, I don't think they do much of that, but I don't know. Maybe they'd be interested. I think they'd definitely be interested if we had a validated assay.
I'm sure.
Yeah, I mean, like if you guys could find this just from your data, it just looks like it'd be so transformative in the blood. You know?
Yeah. I agree.
Totally agree.
But we're trying.
And like you said, it's
Right.
not easy.
Not easy. So it's definitely a fishing expedition.
This is great. Yeah, it's really thought-provoking.
Thank you. Yeah, thank you so much for all your questions and the great discussion. It's good to hear that there's enthusiasm for it, because yeah, we'll see if we can get the funding, but
Great. Okay. I think, Bonnie, shall we end?
Yeah, so I think we're done. Yeah, thanks again for a great presentation. Well, probably we'll have you also soon. So yeah, thanks. We'll probably collaborate with you actually.
Yeah.
We're trying to build
So-
here also in South Texas. Dr. North actually, Scotty North is she one really of the main spearing heads for the proteomics in the brain and genomics. So yeah, we're
Cool.
looking to collaborate with you. Yeah.
Yeah. Please follow up. Happy to chat more.
Perfect. Thank you. Yeah. Thanks all for joining. Thank you.
Thanks, everyone.
Bye-bye.
Thank you
Bye.
so much.